Meanwhile, we're going to move to our second speaker. Minye Zhan is a postdoctoral researcher at NeuroSpin in CERM in France, studying the representation of written words and other object categories together with Stanislas Zhan. And the fMRI study that she will present us today was actually performed during her PhD at the Maastricht University. Please, Minye, the floor is yours.
Okay, so thank you everyone for coming to my talk. So today I'll just go through this study, which was performed in Maastricht University. Let's start from imagining some situation, let's say 1 year ago. Let's say you're attending a conference in person, then you're walking to a conference hall with a lot of people. Then you see someone like this far away. You can't yet figure out the faces because it's quite small, you can't see. So you may start thinking a lot of things. First, maybe you will think, who is she? Do I know her? Then you decided you really don't know her. Then is she waving towards me or someone beside me? Then maybe it's someone beside me. Then you may think that she seems to be quite happy and maybe she met someone she knew. So these thoughts, of course, they are very different if they are coming from you or from someone standing beside you who she's waving from and who knows her. So we can see that the body postures contain a lot of rich information, which include the identity and the action and the emotion and the intention and so on. And we are interacting and reacting to them on a daily basis. So how do we actually understand other people's bodily actions and emotions? To approach this question, in this 7 Tesla fMRI experiment, we used a large stimuli set with 10 predefined categories, which are 80 stimuli split into 2 sets. So we collected the subjective reports from the participants and used the representational similarity analysis, RSA, to study the behavioral representations and the neural representations of these subjective reports. So let's have a detailed look of the categories. So we have 10 predefined categories of bodily actions. In the first 6 categories, they are those daily life actions which doesn't convey emotional information. For example, combing the hair, drinking water, opening door, etc. Or we have also 4 conditions which conveys emotional information, including fear, anger, happy, and sad. So we have 10 participants who passively viewed these stimuli in a scanner. You can see that this is the brain data, actual brain data, and this is the coverage. So we didn't really have the whole brain. We covered most of the brain. And then after the scanning session and outside the scanner, each participant gave individual subjective reports of their perceived action and perceived emotion for each individual stimuli by freely typing in the response dialog. So I'll show you 2 examples of this specific stimulus here. For subject number 3, the answer was cheering for the action and being happy for the emotion. And for subject 5, you see that it's yawning for the action and relaxation for the emotion, which makes sense. But we can see that the subjective understandings are different for some stimuli across people, as we usually observe in daily life. So we analyzed the subjective reports by word embeddings and RSA. For word embeddings, we used the deconf word vectors, which linked the word2vec vectors to the WordNet database, which separates the individual meanings of each single word, because most of the time we have multiple meanings per word. So I'll walk you through the process of our analysis by this example. Let's say we have these 2 stimuli, and we have subject 3 reporting cheering for this one and staring at the ground for this one. So we first reduce these reports into their dictionary forms like this, and then we look up their WordNet senses in the database, which are these. Then we look up the deconf word vectors, which is a 300-dimensional vectors like these. So in cases of entries with more than one words, we average the vectors within the entry. Then at the end, then we do a pairwise dissimilarity computation between these entries. So in this way, we get the perceived action matrix for this subject. And these are arranged by the individual 10 predefined action categories. So here blue means similar and red means dissimilar. And in the same way, we get the perceived emotion matrix for this subject. So looking at the group average matrix, we can see that they largely corresponded to the predefined action and emotion models, as you can see the similarity here. But we can see considerable individual variability if we look into individual matrices here, and their correlation to the predefined models. And the correlations are not particularly high, which are around 0.3 or 0.5. And since all our reports are now in the same common word vector space, we then put them together and ran PCA on all the subjective report entries. So here are the 400 entries of them. And you can see when I plot them out like this by their dissimilarity matrices, you can see the emotions, it's quite okay. You still see the similarity here. But for the actions, because of the individual differences, it's quite, well, it doesn't look really like this. So for the PCA, the first 10 PCs explained more than 86% of the variance for both of these perceived models, perceived emotion and action emotion representations. And looking at the first PC, which are very big in these two cases, they actually didn't really correspond to those categories. If I plot them out by the categories, by different color, you don't really see a categorical structure. However, when we look at smaller PCs, for this case, for action, the 7th and the 9th, they started correlating with the action categories, and you see some action structure here. And they also correlated with the emotions. And for the perceived emotion, you can see that the number 1 and 2 PCs, they correlated more with the emotion, predefined emotion model, and the number 2 and 3 PCs, they correlated with the action and the emotion also, and also the valence. And then we can also plot them in a different way, like in dissimilarity, for dissimilarity matrices for the same data. So we can see that the first PCs, again, they don't really look like, they don't really correlate to this one with a very high correlation. But when looking at the very small PCs, if you squint a little bit, you can see the similarity of this to this. And for the later PCs to the actions. And for the perceived emotions, it's a bit better. You can see the first 2 PCs were correlating with this one quite well. And in the smaller PCs, you can see that PC3 correlated with both the happiness and the valence. And for PC4 and 6, you can see a little bit of the fear, specific fear condition and the sad condition. So for these behavioral representations, we can see that both of these representations were high dimensional and they could not be reduced to predefined models. And then we searched for these specific representations in those individual brains with their individual subjective reports. We found 2 of the clusters, each corresponding to these individual representations. One in the left middle superior temporal sulcus, and the other in the left dorsal premotor cortex. So what are the functions in these regions? Because we don't know what's the first PC doing, and we cannot directly know this answer in the study. So we ask a different question instead. So what are their upper lower stream areas when they're perceiving the actions and emotions in the scanner? By this, we have 2 assumptions for these areas. First, that they may be functionally connected to these seeds. For this, we can use task-residual functional connectivity to check. And for the second assumption, that they may have similar neural patterns to the seeds. For this, we can use the hierarchical clustering to check. And for we found 2 different networks here connecting to the 2 seeds. And the colors of these patches are represented by their dissimilarity to the seed region here. And the more dissimilar, the more red, and the more similar, the more blue. And you can already see that a lot of these regions are a part of the action observation network. This is not very surprising because the task itself is observing the action and emotion in the scanner. Then we plotted the clusters which are the most similar to the seeds and see their overlap. Then we saw that they overlapped in the semantic network. So our study actually indicated that both the action observation network and the semantic network were both involved in the action and emotion understanding. So in summary, we found the perceived action emotion representations, they were both high dimensional and could not be reduced to the predefined categories, but still the smaller PCs from those representations were correlated to the predefined categories. And also in the brain, we found two clusters and found the involvement of both the semantic network and the action observation network. So thank you for your attention and feel free to reach out and chat with me. Thank you.
Thank you very much. Thank you very much. Great talk. Very interesting results. I might start with a couple of questions while we wait and see
Yes.
what gets typed away by the attendees. I was wondering whether you think that some of the overlap with semantic network could be driven by the subjects that have the tendency to name the action that they are seeing. So like they're coming up with the lexical label, so then can lead to an activation.
Yes, I think the lexical content of the response, it's indeed a part of the function of the semantic network. However, well, this is also a part of our understanding, let's say. When we understand one action, we tend to name them, or well, actually in real life, we're not really naming the actions, we're just responding to them. So we are just using these responses as one proxy to understand the understanding process. Because otherwise, as the traditional way of doing the research, we usually use the predefined categories, which are defined by the scientists, not by the participants. And in our case, we show that they are different. There are very big inter-individual differences to the predefined actions. So we are just using one proxy, and there might be better proxies, but what are they? It's not yet sure.
Yeah, no, absolutely. I was particularly interested in that effect of the individual differences. We do have a question from Regina Zaghi. She's asking, did you use different age ranges? Were they all adults? Just wondering, because kids and teenagers might have very different vocabulary for what they for how they interpret others' actions, right?
Yeah. In this case, we recruited participants from our campus. So they are all adults, but we have a quite diverse participants because our university was very international. So we have participants from India, from Greece, from America, and of course Dutch and Germany. So they are not really like a very limited range of participants, but the experiment is conducted in English.
I see. And do you think that would you expect to see similar results if they had been performing a given task over those stimuli? So they were passively viewing, right? Do you think if they had
I think some kind of... Yes. So let's say if we have a different task, which is not passive viewing, but let's say actively judging the emotion, I think the representations may be different and may be modulated by the task. But let's say this is not really a naturalistic way of understanding the emotion, because usually you are not judging a posture when you see them far away. You're actually quickly reacting to them even without thinking. So we are trying to emulate this more naturalistic way of thinking about emotions or observing the emotions. That's why we refrain from an active task. Yes.
Makes a lot of sense. We have another question from Na Eun O. Very interesting. What is your explanation for the first and the second principal components of the actions? Maybe it represents individual differences in the description of the actions, such as the intentions, the situation, the results.
Yeah, it's quite likely, because we can see that because these are arranged, ordered by the actions, then you can see that it's 10 participants, and you can see a quite reliable structure in each cell for each action. So yeah, when you see them overall, they are surely a lot of individual differences, but there is also some underlying patterns. So I'm not really sure what was the PC1 and 2. And if there are better ways to study them, it would be great, but not yet. I do not yet know what they are, the better method to study these. Yeah.
Yeah, it's really not easy. Have you thought of, or would you like in the future to try with even more naturalistic stimuli? Already your images are very, like compared to the usual, the traditional images are very natural, but have you thought of like seeing those action in an environment, in an ecological environment?
Yeah, if it's one option, then it would be great. But the problem was more related to the acquisition of the data, because here we showed each stimulus 12 times, each single picture 12 times to have a very stable estimation of the activity in the brain. So let's say if without this limitation, yes, of course, we will do it. But it will be a very expensive scan with a lot of different sessions for the same participant. So yeah, if possible, yes, of course. Yes.
Thank you. Thank you very much for the great presentation and for answering to all our questions.
Thank you.
We can now move to our third